Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has three main goals. 1. To help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. 2. To build a supportive community where we can exchange the most valuable experience-based research advice. 3. To share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Imagine this. A critical part of your body's defense system, absolutely essential for telling friend from foe, just hidden for decades.
Yeah, it's quite something. Scientists had figured out B cells, how they fire off antibodies.
Right, the antibody missiles?
Exactly. But the T cell receptor, the part that lets T cells actually see the enemy, it was a complete enigma. Immunology's most wanted, you could say.
And totally invisible. Until 1984, when this paper just flipped everything on its head.
It really did, and the big surprise. The T cell receptor wasn't some radically new invention by nature.
No, it looked incredibly similar to the very antibodies they already understood from B cells. Talk about counterintuitive.
And that's the fascinating part. It wasn't just solving one mystery. It unified the whole adaptive immune system. Suddenly, B cells and T cells seem to be working off the same elegant molecular playbook.
Like finding out two completely different instruments were playing variations on the same core melody.
That's a great way to put it, a shared logic.
So what does this actually mean for us? And how on earth did they find it? Today, we're doing a deep dive into that exact paper from Nature, 1984. Sequence relationships between putative T-cell receptor polypeptides and immunoglobulins.
We're going to unpack how they cracked the code, the problems they faced, the really ingenious methods they came up with.
And the impact. The huge impact it had then, and honestly still has today, in medicine and immunology.
Okay, so setting the scene a bit. Early 80s, immunology was, making huge strides in some areas.
Definitely. B-cell immunoglobulins, the antibodies researchers had really nailed down the genetics there.
Yeah, they understood how the genes rearranged VDJ recombination, right? Shuffling these genetic pieces.
Like Lego blocks.
Exactly, like Lego blocks to create this almost unbelievable diversity of antibodies. It was a real molecular triumph. It explained how we fight off so many different bugs.
But then you look at the T-cell side.
Oh, completely different story. Total frustration zone. Everyone knew T cells were critical, killing infected cells, directing the immune response.
The field generals.
Right. But the actual receptor, the thing on the T cell surface that binds the antigen, still missing.
They tried purifying it biochemically.
Repeatedly.
Yeah.
Failed every time. And the genetic approaches, like cDNA hybridisation, they kept getting drowned out by all the antibody messenger RNA, which looked, kind of similar.
So it was like searching for a specific key in a giant box full of keys that looked almost the same.
Precisely. A massive signal-to-noise problem.
But there were clues, weren't there?
Yeah.
Genetic hints suggesting maybe it wasn't that different.
Oh, absolutely. Both T cells and B cells do clonal selection, the idea that the right cell gets picked and multiplies. That functional similarity was a big hint.
Pointing towards maybe a shared genetic mechanism underneath it all.
And the structure, people thought the receptor might have variable and constant regions, the V and C domains that looked a lot like antibodies too.
A family resemblance, but no smoking gun. No actual molecule identified.
Exactly. The molecular link, the specific T cell mRNA that belonged to that immunoglobulin superfamily, that was the missing piece.
And without that piece.
Well, fundamental questions were just speculative. How do T cells learn self from non-self in the thymus? How do they recognize antigens presented by MHC molecules, those cellular ID tags?
Couldn't really build solid models without knowing what the receptor actually was.
Right. So finding it wasn't just, curiosity. It was about understanding T cell specificity, MHC restriction, basically completing the picture of adaptive immunity, bringing B and T cells together conceptually.
Okay, so enter the Davis lab. Mark Davis, Stephen Hedrick, Ellen Nielsen. How did they succeed where others had hit a wall? They needed something clever.
Oh, it was more than clever. It was brilliant. Their strategy, subtractive cDNA hybridisation.
Okay, break that down. Sounds complicated.
Think of it like this. You have all the genetic messages, the mRNAs, from a T cell. That's library 1.
Got it.
Then you have all the messages from a B cell or other cells that aren't T cells. Library 2.
Yep.
Subtractive hybridization lets you compare the two libraries and physically remove everything that's present in both.
Ah, so you subtract the common stuff.
Exactly. You subtract the noise, the shared genes. What you're left with is enriched from messages found only in T cells.
The two-cell specific transcripts, including, hopefully,
Hopefully, the elusive T cell receptor mRNA, it was a way to massively boost the signal for what they were looking for.
like filtering out all the common words in two books to find the unique dialect in one of them. That's smart.
Very smart. And laborious, but smart.
So they did this subtraction. They got their enriched T-cell messages. What did they find? Was it the jackpot?
It really was. They cloned and sequenced what was left, and boom! 2 polypeptides popped out, T-alpha and T-beta.
T for T-cell immunoglobulin-like, maybe.
Well, putative T-cell receptor initially, but the sequence, that was the real aha
Yeah.
moment.
What did the sequence show?
It showed domains strikingly similar to immunoglobulins, a signal peptide, the variable region framework with those telltale conserved cysteine amino acids, a constant region. It looked like an antibody gene family member.
Not just a vague resemblance, but like specific shared features.
Undeniably, a strong family resemblance. The structural hallmarks were there.
Okay, finding sequences that look like the receptor is huge. But how did they prove it? Prove this wasn't just another interesting T-cell protein?
Crucial step. They needed validation. And they used 2 killer lines of evidence.
What was the first one?
Lineage specificity, they checked. Were these T-alpha and T-beta mRNAs present in T cells? Yes, in every T cell clone they looked at.
Okay, and in other cells?
Completely absent. Not in B cells, not in macrophages, not in fibroblasts. Only T cells made these messages. That's a very strong indicator.
T-cell specific.
Yeah.
Makes sense. What was the second line of evidence?
This one was arguably even more definitive. They looked at the DNA itself, the germline DNA in T cells, using southern blots. And they saw rearranged gene segments. The DNA wasn't in its original configuration. Pieces had been cut and pasted together.
Just like?
Just like VDJ recombination and antibody genes, the hallmark of generating immune receptor diversity.
Wow. So these genes didn't just look like antibody genes in their sequence.
They behaved like them. They rearranged in the T cell genome.
So structural homology and the same kind of gene shuffling mechanism. That sounds pretty conclusive.
Exactly. That combination was the killer evidence. It showed T cells use an immunoglobulin-related gene family, one that rearranges, as their antigen receptor. It was this elegant design validation leap that just blew past all the previous attempts that focused only on the protein.
Molecular genetics triumph, really. Solve.
Fundamentally solved, yes. The molecular identity was found.
Now, you mentioned this didn't happen in isolation. What about the environment, the collaboration? What was the secret sauce that helped the Davis lab get there?
Right. Breakthroughs rarely happen in a vacuum, and here the synergy was palpable. Stanford's microcloning facility, absolutely essential for making those complex cDNA libraries quickly. That was a technical advantage.
And the people around them.
Huge factor. They had lots of provocative discussions with Leroy Hood's group nearby, who were experts in antibody sequencing. That interaction really sharpened their thinking about comparing sequences and spotting those IG-like features.
So bouncing ideas off experts in the related field.
Definitely. And having the right materials. They got access to various monoclonal T-cell lines from other key labs, Merrick Kapler, Allison.
Which gave them the diverse RNA they needed for the subtraction.
Exactly. It was this mix of immunogenetics, molecular cloning, early bioinformatics, all coming together across different labs on campus. That kind of interdisciplinary power really sped things up.
It really shows how collaboration accelerates science. What about the individuals on the paper? You mentioned Davis, Hedrick, Nielsen.
Each played a really critical part. Mark Davis, as the PI, made the conceptual bet, really, that the TCR had to look like IG genes, and he set up those crucial collaborations for the cell lines. A bit of a gamble, but based on strong intuition.
And the others.
Stephen Hedrick, the grad student, did the incredibly tough bench work, the subtractive hybridization, screening the libraries. Apparently, there were a lot of false positives. It took real persistence.
That sounds grueling.
It does. And Ellen Nielsen, the postdoc, wielded the sequence alignment tools, pretty basic back then, but she had the insight to recognize those IG-like patterns. Others might have just dismissed them as random similarities.
So leadership, persistence, and computational insight. A perfect team.
Pretty much. Each brought something essential to the table.
So let's talk impact. How did this discovery really change immunology? What did it mean for the field back then, and how does it resonate today?
Well, it was truly paradigm shifting. On a theoretical level, proving T cells use these rearranging IG superfamily genes, that cemented the whole idea of a clonotypic TCR one receptor type per T cell clone, and it unified B and T cell recognition. Same basic molecular strategy.
Completed the picture of adaptive immunity.
Exactly. And technologically, it validated subtractive hybridization plus bioinformatics as a powerful way to find lineage-specific genes. People still use variations of that approach.
And the practical applications, the stuff that affects us more directly.
Huge. Those cloned TCR sequences became essential tools, probes. Scientists use them to map out the TCR genes on chromosomes, understand how T cells develop.
Gnostics too.
Yes, crucially for diagnosing T-cell cancers like leukemias by looking for abnormal TCR gene rearrangements and maybe the biggest impact down the line.
CAR T cells.
Precisely. Understanding the TCR structure was absolutely foundational for later engineering chimeric antigen receptors, the CARs that are used in CAR-RT therapy, a revolutionary cancer treatment.
Wow. So the path from this basic discovery to life-saving therapy is quite direct.
It really is. Now, the 1984 paper didn't answer everything, of course.
Right.
They inferred the protein, didn't directly show it, and how the alpha and beta chains paired up or signaled came later. But it provided the molecular identity. That was the trigger for everything that followed in modern T-cell biology.
What an incredible arc. From this deep mystery, this biological enigma, to something so fundamental to our understanding of immunity and, ultimately, to treatment saving lives today.
It really revealed this beautiful underlying unity, didn't it? A shared molecular language between B and T cells.
It really did.
Yeah.
And maybe that leaves a question for you, our listeners, to think about.
Yeah, how many other biological puzzles out there, things that seem really complex or unique right now, might actually be solved by looking for these kinds of unexpected connections, shared designs across different systems?
What's the next elusive receptor? The next hidden molecule waiting for a clever technique? And what surprising connections might that discovery reveal about how life works?
Something to ponder.
Definitely. We hope this deep dive gave you a fresh look at a truly pivotal moment in immunology. Until next time, keep exploring those surprising connections.